the questions for the Meet the Expert session. I'm going to switch to Dr. Paul Tam Kwong Ang. He's the Li Shu Pui Professor in Surgery and the Director of the Lee Dak Sum Research Center in Hong Kong. And he's the Chair of Professor of Pediatric Surgery there, the Hong Kong University. And he has been Chair of Pediatric Surgery at the University of Hong Kong since 1996. And he's Professor in Surgery, Li Shu Pui Professor of Surgery since 2013. Professor Tam graduated from the University of Hong Kong in 1976 and he worked in the Department of Surgery from 1986. And he was senior lecturer at the University of Liverpool and then reader and director of pediatric surgery at the University of Oxford from the 90 to the 96. And he has numerous awards. He was awarded the 2017 Dennis Brown Gold Medal, the highest award of the British Association of Pediatric Surgeons. And he has been named the recipient of the prestigious Reuben Medal for 2020 by the European Pediatric Surgeons Association. And he has led a number of projects in developing pediatric surgery, especially in China in the past 2 decades. And he has also nurtured a generation over 2,500 pediatric surgeons in China through a training program with far-reach impact. His talk will focus on the tension on the gene mutation in ciliary dysfunction, novel disease mechanism. Please, Dr. Tam, welcome.
Yeah, thank you. I'm very pleased to be able to participate in this symposium. You have my recorded.
Yeah, I'm gonna.
So
then,
you can,
yes.
You can start that, and I'll be available online for Q&A. Thank you.
Of course. Let me just share the screen.
Good day, chairpersons, colleagues, friends around the world. I would like to first thank the symposium organizers for the invitation to speak at this first virtual symposium on hepatobiliary disorders in children in 2022, with the focus on biliary atresia, a condition that is of great interest to all of us. Biliary atresia is a progressive fibroinflammatory obstruction of the bile duct, resulting in reduced bile flow in neonates. It has a very poor disease outcome, with over 40% of patients developing end-stage liver failure, and is the most common cause of liver transplantation in children. We know that the disease probably develops from sometime around birth, with a progressive inflammation postnatally, resulting in the clinical presentation. And it was only the introduction of Kasai procedure that allowed some form of jaundice relief for a number of children. Nevertheless, many will continue to progress to develop fibrosis of the liver and liver failure. There are several interesting features that I would like to highlight. First and foremost is the higher incidence of this disease amongst Asians, with some 10 to 20 per 100,000 in Asians versus 5 to 7 per 100,000 in Caucasians. Conversely, the percentage of syndromic patients appear to be relatively low amongst Asians compared to Caucasians. The etiology of the disease remains poorly understood, but the key pathogenic factors are believed to involve both genetics and the environment. with injury perinatally and genetic susceptibility in the morphogenesis of the biliary tree, both playing important roles. We propose here that perhaps the genetic basis deserves more of our attention than have been in the past. In presenting this talk advocating a genetic basis for biliary atresia, it reminds me of parallel experience of another developmental condition, namely Hirschsprung disease, for which I have been involved in research for over 30 years. While there had been general acknowledgement that the Hirschsprung disease was or is a developmental disorder, sometimes it was viewed that the genetic basis was confined only to the familial and the syndromic cases. It took a very long journey for us to finally come around to understand that the genetic basis of Hirschsprung disease is really important, not just for a small number of patients, but for a much larger number of patients. But we cannot view it as a single gene disorder. We have to realize that the genetic basis we are talking about is not based on the premise that genetic mutations alone is necessary and sufficient for disease causation. While this may be true for a few rare cases, in most of the time, we will find that the disease is actually heterogeneous, and that there are a number of genetic factors which are necessary but not always sufficient on its own to result in the phenotype. In our first effort to advocate this genetic susceptibility pathway of biliary atresia, we undertook a genome-wide association study some 10 years ago and showed that common variants reducing expression of ADD3 and encoding a cytoskeleton protein, adducin-3, increased risk of biliary atresia. And that as we further analyzed the genome-wide association study data, we also discovered that rare copy number variation enabled us to identify enrichment of genes in the immune inflammatory pathway being affected in biliary atresia patients. Fast forward 5 years later, we were able to work together with Professor Mark Davenport to advocate that while in a review in Lancet, that while overall the etiology of biliary atresia remains unknown, we have a framework upon which we can perhaps label our patients accordingly as to whether they would be primarily developmental in origin or occur in isolation, and how genetic factors and epigenetic factors could affect the developmental spectrum and versus the postnatal factors in the viral inflammatory and toxin etiology. Fast forward another 5 years, we now believe that we are nearer the goal of further stratifying patients by pathways, and that knowing the pathways of individual patients may allow us to develop new diagnostic and therapeutic approaches. We presented this review in recent publication in eBiomedicine, as shown on the right lower-hand corner. In order to further clarify the role of genetic basis of biliary atresia, we sought to complement our original study of the common regulatory variants predisposing to biliary atresia identified in our GWAS study with a study on rare coding variants that normally would be associated with syndromic biliary atresia, such as polycystic kidney disease L1 gene. And we hypothesized that some of these rare coding variants may also predispose to non-syndromic patients. With this, we hope to identify novel genetic factors associated with risk of biliary atresia, and the rest of the talk will focus on this study, which was published in eBiomedicine in the article cited here, the right lower-hand bottom corner. In this study, we whole exome sequenced 89 non-syndromic biliary atresia trios, meaning that we studied not just the biliary atresia patients, but also their unaffected parents. The approach of whole exome sequencing allows us to detect rare coding genetic variants across the entire genome, and that by filtering for rare damaging variants, we would be able to focus on possible causative genetic mutations. This we achieve by filtering with the inheritance pattern, focusing on de novo and biallelic variants. We then will filter these through in silico deleterious prediction, and then by means of filtering with accepted allele frequency and gene expression in liver, we would be able to focus on rare damaging variants that are expressed in liver and may therefore play a pathogenetic role in biliary atresia. So this is the schema of our study, where we have the filtering criteria, allowing us to identify 239 genes with rare damaging protein altering variants of larger effect. With this, we tried to enrich the biological significance of these rare damaging variants through gene ontology and enrichment analysis. Here you can see that the genes group very nicely into several major pathways, of which the focus today is on cilium, microtubule associated organization, binding, and movement. There will be other pathways of interest, such as extracellular matrix organization and regulation, which we will have to define further with future studies. But what is amazing to us was that in the analysis of the pathway and enrichment analysis, 7 significant biological processes involving 49 genes were identified. These biological processes include cytoskeleton organization, cilium organization, microtubule cytoskeleton organization, microtubule-based process, spindle organization, organelle assembly, and cilium assembly. And half of these genes are ciliary genes. Now, we have to understand that cilia is widespread in the body, but in human cholangiocyte, cilia is non-motile and serve as a mechanosensory function and calcium influx. So how does it affect the process of bile flow? We believe that the primary cilia can detect bile flow and regulates the Wnt, hedgehog, and notch signaling pathways. And that when we analyzed the ciliary mutations in our patients, we found a 2-fold increase in the mutational burden compared to controls. Most importantly, 31.5% of biliary atresia patients carry more than 1 liver-expressed rare damaging ciliary mutations. And there's a high level of locus heterogeneity in non-syndromic biliary atresia involving diverse ciliary functions and localization. This ranged from the basal component, TTC 17, to cilia assembly and PCNT, all the way to intraflagellar transport of KIF3B. To further clarify the pathobiological implications of ciliary mutations in biliary atresia patients, we studied the mutant BA livers and found that cilium exome were absent in all the bile ducts of liver biopsies of mutant biliary atresia subjects. Here you can see that in the control, the co-localization of the cilia staining of both the PCNT and tubulin, whereas in mutant biliary atresia, these were absent. As to the possible mechanism, we observed that there is a dysregulation of PET1 and GLI1 in both the mutant BA liver to suggest a correlation between the abnormal cilia and the aberrant hedgehog regulation in these biliary atresia patients. Obviously, to define further the implication of the hedgehog signaling will require more detailed study subsequently. We also showed that there was a significant reduction in the number of cilia in human fibroblast cells. You can see that in the control human fibroblast cells, there's a high percentage of ciliated cells, whereas in those experiments where silencing of the relevant ciliary genes were carried out, the percentage of ciliated cells decreased dramatically. Cilia were detected in some 0 to 12% only of human cholangiocytes with TTC 17 and KIF3B. siRNA compared to 50% for scrambled siRNA by scanning electron microscopy shown here. So in two separate studies, both in human fibroblast cells and in human cholangiocytes generated from our study with organoids, which I will refer a little bit later on, you can see that the silencing of the ciliary genes will result in reduced number of ciliated cells. What about its effect on in vivo? We further performed gene knockout experiments in zebrafish and showed that while there was no gross biliary developmental anomalies, there was defective bile flow in mutant zebrafish embryo, as shown here. You can see the bright staining in the wild type compared to the much reduced luminescence in the mutant zebrafish embryo. Finally, we sought correlation clinically. We asked the question as to whether the ciliary mutations observed in biliary atresia patients also affect other parts of the body in these patients. And we carried out ciliary function tests on patients for whom we could recall. We were able to recall 10 of our studied patients who carried the deleterious ciliary mutations. We performed the nasal nitric oxide test, which is widely adopted as a screening test for primary ciliary dyskinesia. And you can see that some 8 out of 10 of the biliary atresia, the mutant biliary atresia patients had low nasal nitric oxide levels, for whom 4 are in the range of the primary ciliary dyskinesia, and 4 are in the lower end of the or beyond the healthy range. We also studied the ciliary function with both electron microscopy and high-speed video microscopy, and found that whilst in control, you can see the very nice coordinated frequent flow of the cilia. In the BA patients with ciliary mutation, the ciliary frequency is lower and also appear to be less coordinated. So in summary, we are able to show that ciliary gene mutations leading to cholangiocyte cilia malformation and dysfunction can be a key biological mechanism in biliary atresia pathogenesis. We propose that genetic factors play a major role in a substantial proportion of non-syndromic biliary atresia cases. A subset of patients with ciliary mutations may have potential pleiotropic phenotypes, and genetic screening would allow patient stratification to further assess the subclinical features and to improve clinical management. A follow-up study on clinical features and long-term outcome of these patients are ongoing. So how else could we unravel the mystery of biliary atresia? We also undertook an important study utilizing liver organoids and showed that there is beta amyloid deposition around liver bile ducts, which suggests that this could be a part of a novel pathobiological disease mechanism, and may also provide a new diagnostic tool for biliary atresia. This study was published in Journal of Hepatology in 2020. So where do we go from here? Our group firmly believes that the biliary atresia can have an improved treatment outcome through the translation of disease mechanism discoveries. And this would involve both the genetic stratification and discovery, and the development of intervention and biomarkers based on these new knowledge. So of relevance to the genetic study, we are currently undergoing a larger cohort of study of genomic signatures of BA trios, coupled with omic studies to identify molecular signatures of these patients, and hopefully to allow us then to segregate patients into those who have a good prognosis versus those who are destined to have a bad prognosis. And by stratification of subgroups through genetic etiology, we hope to devise a stratified precision medicine approach for biliary atresia. This is how we envisage that with the identification of genetic risk factors, we would both understand better the biology of the disease and improve the genetic risk stratification, leading on the one hand to novel alternative therapy to surgery alone, and on the other hand for more precision diagnostic and therapy, thus improving overall clinical management. So with this, we hope that we can alter the course of these poor babies suffering from biliary atresia who after Kasai surgery suffer from various problems by the discovery of new biomarkers and intervention, thereby reducing the need for liver transplantation through the intervention of these therapeutic windows. I'd like to acknowledge all my colleagues who have contributed to this study, both locally and globally. And I would invite those who are interested in this endeavor to join us in our group as postdoc fellows or as PhD students. Thank you very much.
Thank you very much, Dr. Tan. That has been a wonderful talk. And now it's time for questions. I have a first question. If I don't see any other question, the people is starting their questions. I wanted to ask you, in the trios you have a study, or in the families of these genetic biliary atresia kids, do you see how is the penetrance of these mutations, or have you see other types of biliary tree abnormalities or mild phenotypes, or maybe other consequence of ciliary dysfunction?
Thank you. Very important question. In our patients, we have to remember that they are firstly syndromic, non-syndromic patients. Secondly, that the findings identified a group of genetic mutations all of which can be mapped to ciliary dysfunction, but that they are not the typical monogenic disorder caused by a single gene mutation. So they are otherwise normal. They do not have syndromic features or non-biliary atresia phenotype. As we discovered the ciliary mutation, we then recall the patients for study of ciliary functions elsewhere. The most common ciliary function dysfunction relates to the airway, and that's why we chose to perform the nasal nitric oxide test. And because of COVID-19 and the nature of the study, we were actually fortunate that we were able to recall 10 out of the 89 patients, remembering that only about 29 of these patients had ciliary mutation. So we recalled 10 out of the 29 patients who demonstrated the ciliary genetic mutations, and showed that they, although they did not have clinical manifestation of respiratory disorder, they showed ciliary dysfunction using the nasal nitric oxide test. So I believe that they may either be subclinical in their non-liver phenotype, but they may develop the respiratory problems in later life, and therefore we would have to follow up in future these patients more long-term, and not just in isolation on the liver function, but to be aware that there could be more widespread pathology. Thank you.
Thank you very much. And Dr. Carpen was asking a similar question. So if there's any extrahepatic features of those biliary patients with a stronger ciliary gene footprint.
I don't know whether this is more an Asian feature, or whether this the findings would be replicated in a Caucasian population. So hopefully through sharing our findings in this global symposium, we would stimulate other researchers around the world to have a look at the genetic and the ciliary aspect in biliary atresia.
Really good.
I have a question, Dr. Tam. That was a beautiful talk, really interesting. So I was wondering if studying the family history of those patients, you find maybe, I don't know, some long-term familiarity or somebody that has biliary atresia or something like that, because if you are finding these strong ciliary gene mutations, it's expected that maybe other members also have, maybe not with the same severity, but.
Yeah, I think that would be ideal, that if we could study the parents and the siblings as well, to see if there could be hidden ciliary and or respiratory problems. But no, this is a little bit of a surprise finding for us when we did the whole exome sequencing as a bias-free approach and identify such a high incidence of ciliary mutation. And I think that's due to the fact that we were able to focus on biological process rather than looking for a single gene.
I'm very intrigued about your last slides were very interesting and very exciting. I hope you can get all these samples and do all this work, which is also expensive, and and you have to. But I'm sure you will. And I wanted to ask you. You put there the low pass full genome sequencing. Are you thinking on what sort of data are you waiting for? If that's something that you can say, what
Yes.
sort of data are you expecting of those analyses?
Well, it's a 5-year project, so I hope to be able to share more of the findings with this group in coming years. But the idea of genetic study is that you would gain more power with more patients. So number one is to recruit more patients. Number two is, I believe in integration of the genetic findings with clinical findings. I think up till now, perhaps the scientists have been doing the scientists' work and the clinicians doing their clinicians' work, and there needs to be more of the coming together of the data. Is speaking as a clinician scientist, I can say that it is not easy because the clinical data are often missing, and if you have some important information not available in the database, then it makes the interpretation less robust. So we hope that we can overcome this by planning prospectively what genetic data and what clinical data we wish to obtain, and then analyze together. So that's how we're looking at, and we do not think that the genetic study alone will give us the answer. It is a heterogeneous disease, and I think this is really only going to be one of the key features that we should characterize in our biliary atresia patient.
And based on what we have seen today, I think probably you need to do also probably flow cytometry, single cell sequencing, yeah, or TCR sequencing, BCR sequencing. I don't know. There are many omics there, yeah, to because
and,
yeah,
the inflammatory pathway, yeah, just immune inflammatory pathway just shown by the Guangzhou group is also very exciting. So I think it's there are many factors, and it reminds me also a little bit of how the community approached another intractable or incurable condition of pediatric child or pediatric solid tumors. When years gone by, we were just focusing on simple surgery. Then it evolved into a very sophisticated multidisciplinary approach, and the patients improve with these novel and integrated therapy. But even in child tumors, there are some tumors which respond better to treatment, some which do not respond as well, even with the latest advances. So I think this is going to be a long, hard battle, and we need everyone in the community to believe in the effort and perhaps participate more in the collaboration, because otherwise we won't be able to conduct large-scale clinical trials for these rare disorders.
I think we all agree with that, and we hope we can collaborate more and more. I think it's time for the next, maybe, I don't know if there's any other question around. And thank you very much, Dr. Tam. Lucia, Dr. Zhang, because of the time. You know, we know that there is 2 a.m., 3 a.m. now, so we really appreciate your patience and your participation. You know, with more appreciation because of the time of the hour.